recording the meeting, and so we're talking about metabolism. And one place we want to start off, I know we've talked about in the past protein structure and some degree of protein function, and there's another really important protein function that you see a lot in metabolism, and that is with enzymes. And the thing to understand about enzymes is that they're catalysts, okay? And I want you to just tell me what comes to mind when you think of the word catalyst. It's sort of in your everyday vernacular. What do you think of?
Get it started.
Okay, yeah, okay. Okay, all right. So it's making something happen that maybe wouldn't happen very well otherwise? Okay. Any other thoughts?
Like a bridge. Combustion.
Are you thinking about combustion?
Yeah, but she said something and I didn't know she was saying, I think breach, did you say?
I said like, it's like a bridge. It's like it connects two things.
Ah, bridge, yeah. Okay. Fantastic. Okay, yeah. So it's a connecting thing. All right, okay. Do catalysts make things go slower or faster?
Faster.
And one of them definitely is that they make reactions go faster. And it can seem like they're getting them started, actually, because some of the reactions are so slow that it would take them, I don't know, 100 years to go without a catalyst.
Wow.
And so, but they could still go forward. That's, the point is that they're taking a reaction that can happen, but it's just really slow, and it's speeding it up. And then the other thing, well, there's actually two more things with catalysts. They tend to be pretty specific. You don't usually have catalysts that work for lots of different kinds of reactions. Generally, they just work for one kind of a reaction. Okay, so they're pretty specific. And then the other thing about it is they're like really sustainable because they're reusable. Okay, and when we say they're reusable, what I mean by that is they're not changed by the reaction. They help a reaction go forward, but once that reaction is done, they can do the reaction again. So they can do it again and again and again and again. And that's why we usually only need a very small amount of catalyst. And I just realized that my camera's kind of, there we go, okay. A very small amount of catalyst to do a lot of work, a lot of chemical work. And so when we talk about enzymes are catalysts. And the only thing that sort of distinguishes them from just a catalyst in general is that they're made of protein. Okay, so they are protein catalysts. And that's it. Otherwise, they're just like any other catalyst in that respect. Now, because they're made of protein, that makes them susceptible to all the things that affect protein structure, right, that we learned about in task 2. But, yeah, they're catalysts, so they make things go faster. And the way that they do that is actually in a really specific way. If I have some reactants, let's say I've got a reaction where I've got A plus B and it's going to C, okay, just whatever reaction. Here are my reactants. I've got A plus B. And what I'm showing here, this is energy on this axis, okay? It's sort of you can think of it as like the stored energy in a molecule. And here's going to be what we call a reaction coordinate, meaning how far is the reaction gone, okay? And that's going this direction. So if the reaction hasn't started, then I'm over here, okay? When the reaction's finished, then I'm over here. And reactions take place usually because the reactants are higher in energy than the products are, okay? And so thermodynamically, that makes it really favorable for the reaction to happen. But it doesn't always happen just because overall it would be favorable. Sometimes there's a barrier. There can be a barrier to that reaction, okay? An energetic barrier, meaning that to get the reaction to go, we're going to have to put energy in to get it up to this point here, what we would call an intermediate, so that it can then finish the reaction. And the difference in energy between the reactant and where we have to go to get the reaction to start is known as activation energy. Okay, so this, sometimes we call it E with a subscript of A. So this is our activation energy, and you can, This is the amount of energy we have to put in to make the reaction go. And what a catalyst does, it can't change the reactants, it can't change the products. What it can do is lower that activation energy, and that's what it does. So when an enzyme is present, or any catalyst really, what it's doing is it's lowering this activation energy, so now you can see it's a lot easier to get that reaction done. And so it's going to happen much more quickly, okay? And one sort of, more concrete analogy to this would be if I was on a hill and I had a big boulder, right? And the hill goes down. I mean, that boulder wants to go down the hill, right? But if I put up a big barrier here, then the boulder's not going to go anywhere unless I put in a lot of energy to get it up and over that barrier. But if I lower that barrier, I'm going to lower it, okay? So that it's really just, just a little bump. Then I just, give it a little shove, and then down it goes, right? Activation energy works very much the same way. So we're taking a reaction that wants to happen, right? Just like this boulder wants to roll down the hill. But it's how big of a barrier do we have? And the enzyme reduces that barrier. So the reason it makes things go faster is because it's reducing the activation energy. Yeah, so you can see here's my, in red I've got my activation energy with the enzyme, and in black I have my activation energy without the enzyme, right? And I mean, just visually, just looking at it, you can see it's just going to be a lot easier to go over this red one, right? And so it will happen much more rapidly, much more often, okay? And so enzymes, they can be pretty amazing. They can take reactions that in our bodies, just, hanging out in water would take literally like years to do, and they can make it happen in seconds. They can make several of them happen in a second. And so they can speed things up like just so much, millions of fold sometimes, millions of times. So they're pretty amazing. Now, again, just like other catalysts, enzymes are specific, okay? They're really very specific. So when we talk about what an enzyme is working on, so what is this A and B thing? Let me just draw out. I've got A plus B, and it's going to C, and I'm using an enzyme, okay? Then, and I'm going to put enzyme here. And by the way, this is usually how they're drawn in biochemistry. If there's an arrow going from one chemical to another, and there's a name written below the end of the arrow, that's usually the enzyme. Okay, the arrow is kind of representing the activity of the enzyme. The other thing is that enzyme names always end in ASE. So kinase, hydrogenase, carboxylase, like all kinds of aces. If you see an ace, then that's an enzyme. And you've seen some aces before, right, in your nucleic acids task with our polymerase and our primase, right? Those are enzymes because they have that ASE, and they are catalyzing the reaction. Okay, so whatever is at the beginning of the arrow, these are known as substrates. When we say that something is a substrate for the enzyme, that means that's the reactant of the reaction that the enzyme is catalyzing, or that reaction, okay? In our case, it's A plus B. So A and B are both substrates for this particular enzyme. Whatever is at the end of the arrow, this is going to be the product. Okay, or products. It can, take more than one product sometimes. All right. And this is really important to understand, to know what is the substrate of an enzyme and what is the product of the enzyme, because they're very specific, and so it's really just going to have one or two, okay? And when we start looking more at metabolism, then we start talking about pathways. And when we talk about pathways, that means lots of enzymes. So let me just give you an example. If I have A, and it's going to C, and that's going to C. Okay, and I'll just call this, I'm going to be really creative here, 1 ace and 2 ace and 3 ace. Question, what is the substrate of 2 ace?
B. AB. A plus B.
It's just B. It's just B.
Okay.
That's what's at the beginning of the arrow for 2/8. Okay? That's a really, really good distinction. I'm glad you mentioned that. Now, what is the product of 1/8?
That would be B.
The product of 1/8.
That would be B.
Be both a product of one enzyme, right, and a substrate of another. Now, C can get a little complicated, right?
Yeah.
It's the product of 2/8, but it's the substrate for both 3/8 and 4/8. So,
okay, that's not too bad.
Whatever's at the beginning of the arrow is the substrate. Okay, whatever's at the end of the arrow is the product. That's you. But what if I did this? And I gave you 6/8, right?
So it's a product.
Then it's a product of two things, right? And metabolism is a little bit like that. Things get really interconnected. And the thing is, it's easy to look at those pathways and just feel like, whoa, I'm so overwhelmed, right? But just remember, just look at the arrow. Whatever's at the beginning of the arrow is the substrate for that enzyme. Whatever's at the end of the arrow is the product for that enzyme. And so just take it a piece at a time, okay? There might be a lot of pieces, but that's okay. You just look at it a piece at a time, okay? So, I want to do a little bit more pathway practice here. We're going to get ready to this. All right. So let me ask you a question. What if something were to happen to, let's say, 2/8? Okay? What happens if 2/8 isn't working for some reason? Maybe there's a mutation. Well, so this is something that happens in a lot of genetic diseases, like metabolic diseases, where there will be an enzyme that's just not working. If you think about PKU, for example, which is probably something you've heard of, right, with infants who have the phenylketonuria, right? Ketonuria.
Yeah, when they're not supposed to be taking the phenylene product.
Oh, it's because they can't break it down, and the reason they can't is because they have an enzyme that's broken. It's missing. So let's say that happened here with 2/8. Okay, so 2/8 isn't working. What do you think would happen? Like, just give me some intuitive ideas about this. Like, what's going to happen? Is 3/8 going to be able to do its job?
No.
Why not?
Well, if C won't exist, or it'll be not as strong.
Yeah, right. So C is not going to be there unless we have something else that's making C, right? C won't be there, and so all of this stuff isn't going to work either, right?
Right.
No problem.
Right.
Now, what about B? What's going to happen with B?
It's done. I mean, what does it got to do now?
Yeah, exactly. Now, 1/8 is still working, right? So anytime I have A, I'm going to get a lot of B, right? So B is going to start building up. You'll end up with a lot of B. And in the case of certain diseases like PKU, that buildup of the metabolite that can't be broken down is what causes the toxicity. This is what happens. B starts to build up. And so you start to see how metabolic pathways can be a bit like the pipes in your house, right? If I've got, let's say I've got some pipes going like this, right? There's a branching point. If I get a clog here, well, no water is going to go here anymore. And I'll probably end up with extra over here, right?
Right.
If I were to get a clog here, well, then I'm not going to get any water.
Anything anywhere.
Yeah, it's going to get stuck, right? And usually what happens with plumbing, I live in an old house, so I'm very familiar with this, is this joint or whatever it is, it'll start to break, right? So something starts to break. It's just, things aren't good, right? It's not good to get these blockages, these big buildups. And we see that. We see that in metabolism, okay? Okay, I just want to kind of take one quick little divergence here from our pathways and talk a little more about enzymes. So we know that they're specific, right? And
Yes.
when we talk about sort of like the theoretical model of why they're specific, a really popular one is known as the lock and key model, okay? And let me just show you a picture of that here. And we'll move it up so you can actually see it. This
Thank
isn't
you.
your only chance, don't worry. All right, so you can see here, so the enzyme is represented by this gray blobby thing. And you can see that it's got this characteristic shape, and that's where the substrate is going to bind. Now, remember, these are really specific. And so where the substrate binds is what we call the active site. And we call it the active site because that's where the action happens. So, you know, pretty straightforward. And the active site will have a particular shape. It's going to have a shape that's complementary to the substrate. Okay, that's how it can tell that, oh, this is my substrate and something else isn't. Okay, so here's our substrate. Here's my enzyme with the active site. And so this is a situation where the enzyme and the substrate are floating around in solution, but they're not attached to each other yet. Okay, sometimes this is called the E plus S, meaning enzyme plus substrate in solution, but not attached. Then the substrate combined, and you can see it combined because they have this nice complementary shape. And sometimes we call this the enzyme-substrate complex, or just ES. Okay. Now at this point, because the enzyme now has its substrate bound, it can do its magic. It can do its reaction, whatever it does, like the chemical reaction that it's going to do. And in this case, it's severing this connection here. So now it's made a product, and it's got the product inside of it. Okay, so this is what we would call an enzyme product complex, or an EP. And then it's going to let go of those products and let them float away and, you know, go on to their fate. And we call that E plus P, or enzyme plus product. Okay? And the thing to keep in mind, what's important about this model is not only that the substrate and the enzyme have these complementary shapes, but also that the enzyme here at the end is the same enzyme, same shape, as the enzyme here at the beginning, which means it can go back and do it again. So it's reusable. Okay? And so this particular model of enzyme action, the lock and key model, helps us to sort of visualize both the specificity of the enzyme as well as its reusability. Usually what happens in a pathway is 1A is going to take A, turn it into B, let it go. B is going to float around until it runs into 2A, and then 2A will grab it, turn it into C, and let it go. So that's how it happens. And I know that seems really inefficient to us because in human terms that seems like it would just take a long time. But remember, they're inside of a cell. The cell is really small and compact, and things actually move around really quickly inside of the cell. And so most of the time, they don't really need to hand them off. You know, 2H will find B just fine, and the reactions will keep going. Well, each enzyme is, so all of these enzymes are acting at the same time, right? So 1A, it's going to pick up whatever A is around and turn it into B. And 2A, whenever there's B around, we'll pick it up and turn it into C. 3A, the same thing. Okay, you can kind of think of the cell as like a stew, with lots of different vegetables and things in there. And kind of whenever a carrot runs into a piece of celery, something else will happen, right? And that's going to happen. It's just going to happen.
Okay.
Great question.
Now,
I love the connection.
The enzyme does not change its shape, though.
No, the enzyme is the same.
Just the substrate.
As it is at the beginning, right?
Okay.
Exactly. So that's what makes the enzyme reusable. Is it's not any different after the reaction as it was before, and so it can.
I like that. It's reusable. It's reusable.
Yeah. It's one of the reasons that you don't need very much, right? You just need a tiny bit of catalyst to really make things go. And it's another reason why they're so popular in lots of commercial applications. For example, your laundry detergent now almost certainly has catalyst in it, or I should say enzyme, because they only need a tiny bit, and it can help to chew away all of the stuff that gets up on your clothes. And so they're very effective. That's right. We're understanding pathways. Now let's talk about a specific pathway. Okay. Now remember, pathways can look scary, but just take them one piece at a time. All right. So we're going to talk about hereditary fructose intolerance. Okay. And I'm going to show you a pathway. But before I show you that pathway, I want to show you one other thing. Let's talk about sucrose. Now, we know sucrose is table sugar. Okay? And sucrose is made up of a glucose molecule hooked to a fructose molecule. So when we eat some fructose, the first thing our body has to do, sucrose, the first thing our body has to do is actually break those two things apart. And so it does. It has an enzyme that does that. So you end up with glucose plus fructose now. And then they enter their respective metabolic pathways. So now we're going to look at the different pathways they enter in the liver, okay? Keep in mind, most cells in the body run on glucose. They don't do much with fructose. The liver is going to be what metabolizes about 70% of the fructose that we take in. It looks scarier than it really is. I'm making it bigger, so hopefully you can see that. Okay. And, okay. So now, here's glucose. Okay. It's coming in through its own little glucose transporter, and it comes inside the liver. So out here is blood, okay, and here is a liver cell. Okay? And this is after a meal. So I've just had a meal. I've got lots of glucose and fructose and all kinds of things, in my blood. And so the liver is going to take up some of that glucose to help store it for later. Our liver is really important for that, for maintaining blood sugar levels. And if I just let the glucose come in and just keep it as glucose, the problem is that this transporter is kind of like a revolving door, okay? So the glucose can actually just go right back out. And that's not terribly efficient. And so what it does, the liver has a special enzyme, glucokinase. Notice the ACE, it is an enzyme. It's going to put a phosphate on that glucose. And what that phosphate does is it means that it can no longer escape the liver cell. It has to stay there. Okay. And the other thing that it does is it sort of tags it to enter one of two pathways. Either it's going to be stored as glycogen, which is what usually happens, actually, in the liver, or if the liver already has lots of glycogen, or if the liver needs energy, if it needs ATP, then it could send it down glycolysis. So all of this right here, you'll recognize pyruvate here. This is glycolysis. And if you don't recognize pyruvate, you will in just a little bit. I'll talk more about glucose metabolism. So pyruvate, when we go through glycolysis, the body will make a choice on what to do with it depending on our energy needs. It can either make ATP if our cells need ATP, if they need energy. Or if our cell already has a lot of energy, then they'll start to store them. They'll store them as fatty acids, especially the liver. Okay, the liver in particular will store that energy as fatty acids, send them into the blood so they can get stored in our fat cells. Okay. Okay, so we've got these two options, but basically both of them are, energy storage for the most part. The glucokinase, it will do its job as long as it's in the cytoplasm of the cell. So just a quick reminder, here's a cell. It has a nucleus, okay? Anything outside of the nucleus is the cytoplasm. And this is where the glucose is going to be, is in the cytoplasm. So glucokinase can only do its job, I'm just going to call it DK, all right? It can only do its job when it's in the cytoplasm.
Okay.
Since that's where the glucose is, right? There's no glucose in the nucleus. Now, fructose, when it gets taken up, also comes in through a transporter. It has its own kinase, fructokinase, that is also going to put a phosphate onto it. So now we have fructose 1-phosphate. This F1P, that stands for fructose 1-phosphate.
Mm.
And you'll notice that fructose 1-phosphate is the product of fructokinase and the substrate of aldolase B. So just like our other pathway, right, one molecule can be a product of a substrate, right? Aldolase B, and this is an enzyme that only exists in the liver, a little bit in the kidney and small intestine, will essentially chop the fructose molecule in half and make DHAP and glyceraldehyde. And these are actually intermediates in glycolysis, and so they can very easily enter glycolysis to make ATP or fatty acids, or they can actually go the opposite direction. Let me change the color here so it's easier to see. If they go the opposite direction to make glucose or glycogen, then it's called gluconeogenesis. Now, gluconeogenesis sounds like an awful word, right? Like something you would have in a spelling.
Yes.
Yeah. So let's break it down, okay? So gluco
Sugar.
Okay. Neo means
I don't know.
new. It means new. And then
Oh.
genesis means to make or to
The
produce.
beginning.
Well, yeah,
Oh.
right, right. I mean, in Latin, technically, it means to make or to produce, or yeah, that sort of thing. And so, really, this is making a new glucose molecule. That's all gluconeogenesis is. Okay, it's glycolysis in reverse. So, when aldolase B tops up our fructose 1-phosphate into DHAP and glyceraldehyde, they can either enter glycolysis, going to ATP, or it can enter gluconeogenesis and go get stored. Okay. All right. So that's how things are happening. Now, what's kind of interesting is that this fructose 1-phosphate, in addition to being a product and substrate, turns out it's also a signal. And it's a signal to our friend glucokinase up here. And it tells glucokinase, come out of the nucleus. Come be in the cytoplasm so you can do your job. Okay? And this is kind of a shortcut that the body has developed over, thousands of years. Because usually we're only going to have fructose in our blood if we also have a lot of glucose, right? So if the liver is sensing that it has fructose by having fructose 1-phosphate, then it's a pretty good bet that it also has glucose, and so it's going to need to take it up. And so again, it's sort of a shortcut for the liver to be like, oh, there's fructose around, turn on the glucose machine, because, we have it. We know we have it. And when fructose is low, and glucose is also low, so our blood sugar is low, that means I'm not going to have very much fructose 1-phosphate, and so glucokinase is going to go to the nucleus. And why do we want that to happen? Well, let me show you. Okay, so now we're looking at our liver glucose and fructose between meals. So I haven't just eaten, my blood sugar is low. Our liver's job in that case, we know, is to provide some glucose, right? To feed our brain and our other cells before we eat again. And so in that case, well, our fructose is really low, which means fructose 1-phosphate is really low. Glucokinase is in the nucleus, okay? And what that means is that the liver is not going to be taking glucose up, okay? Instead, it can break down its glycogen. It can go through gluconeogenesis, remember just glycolysis in reverse, and it can make glucose and then release it into the blood, okay? Without glucokinase in the way, we can release glucose into the bloodstream, stabilize those blood sugar levels, and the body is happy, okay? That's how things normally work. And they work pretty well, right? They work pretty well. Now, remember our pipe analogy. Now let's think, what is going to happen when, and I guess I got these in reverse. I should have done this stuff in green because it's normal. But anyway, hereditary fructose intolerance. This is a disease we've only known about for maybe 40 years, and let me tell you why. It's awful for people who have it. If they eat something that has fructose, in other words, that tastes really sweet, they feel terrible. And so in the past, they've just self-regulated. They just don't eat sweet things. They just become known as these people that, they just don't like sweets, right? But it wasn't really known as a disease until there was a gentleman in Switzerland, I believe, about 40 years ago, who had hereditary fructose intolerance but didn't know it. He just didn't like sweets, right? They didn't make him feel good. And he was in the hospital for a pretty normal procedure, and they gave him an IV, and because they were trying to be low glycemic index and all of these progressive things, the IV was, or the IV fluid, instead of having glucose, had fructose. Now he couldn't taste it. He couldn't self-regulate. But his body was getting flooded with fructose. So let's look at what happened to this gentleman. He has hereditary fructose intolerance. That means his aldolase B does not work. So if aldolase B is not working, what does that mean for this fructose 1-phosphate? If this is my pipe and I'm blocked, is my fructose 1-phosphate just going to go away? Or is it going to stick around and build up?
Yeah, it's going to build up.
Yeah. Exactly. So we end up with tons and tons of this fructose 1-phosphate in his liver. Okay? And it turns out this has a couple of consequences for man. So one thing is that we see this phosphate, right? Now, normally, fructose 1-phosphate gets made, and then it's broken down, and it goes through all these processes, and that phosphate is eventually used to make ATP. Because our bodies, all of our cells actually require what we call free phosphate. When I make ATP, and we'll talk about this in a little more detail, but when I make ATP, I'm actually taking ADP, which the D stands for di, so it's ADP, there's 2 phosphates on it, and we're adding another phosphate, so that we have ATP, or triphosphate. Okay, there's 3 phosphates on there.
Oh, I never knew that. Try.
That's all it means. Cool. That's all it means. So obviously to make ATP, we've got to have some free phosphate around, right? Well, what they noticed with this man, and then put yourself in this poor nurse's position, the guy had nothing but an IV given to him, and all of a sudden, he starts getting a little yellow in the eyes. He starts throwing up. He starts getting really dizzy. So they do some blood work, and they find liver enzymes in his blood. So he's got some liver damage. Things are happening to his liver. Now, the sad thing is that this gentleman did actually end up dying because they didn't know what was wrong until it was late. But when they did a biopsy of his liver, what they found was that the amount of free phosphate was really, really low. And it was low because that phosphate was getting bound up on this fructose 1-phosphate. It was stuck. And without a lot of free phosphate, the cell couldn't make ATP. And liver cells, or any cell for that matter, that can't make ATP does not survive. Right? And so this phosphate issue was leading to one set of symptoms. This is our liver damage. And then if they keep having, fructose, it will eventually lead to liver failure and death. Yeah, exactly. Okay. Now, unfortunately, that's only half of the symptom. This poor guy. The other part came because fructose 1-phosphate, in addition to being the substrate for aldolase B, right, it's also a signal. Remember? To glucokinase. It tells glucokinase to come out of the nucleus.
To come out of the nucleus, right.
Mm-hmm. Now, let's say my blood sugar is low. I don't have glucose, really, to take up out of the blood. Not very much, anyway. But I've got this fructose 1-phosphate hanging around, telling my glucokinase to come out of the nucleus. So glucokinase, anytime, so the liver is receiving signals from the body saying, release some glucose, we're hungry, right? My brain needs some energy here. And so the liver is trying to break down glycogen, right? It's trying to go through gluconeogenesis here. But whenever it's producing glucose, our friend glucokinase is there, and it puts a phosphate on it and sends it back into the storage pathway. And so what ends up happening is very little glucose is able to be released from the liver cell into the blood. That means we've got chronic low blood sugar, hypoglycemia, with all of its fun associated symptoms. Okay? So the symptoms of HFI are twofold. You've got the hypoglycemia part, because of that connection between fructose 1-phosphate and glucokinase, right? Slowing down gluconeogenesis, it's slowing down glycogen breakdown. The liver cells just can't keep up with the demand of the body for glucose. And then we also have, the poor liver has its own problems, right? It can't make ATP at the same level because its phosphate is being depleted, because it's stuck on fructose 1-phosphate. So the liver cells are starting to fail, starting to die. The ones that are still around have a really hard time releasing glucose, so the rest of the body is hungry, very hypoglycemic. And so as long as that person is still being fed fructose, it's not looking really good. So this is really a disease of the hospital. Because again, most of the people with this disease self-regulate. They don't even necessarily know they have it. They just know they don't like sweets, that sweets make them feel awful. They avoid them. But when they're in the hospital and getting an IV, they don't know what's in the IV fluid. And so if they're and they can't self-regulate. So if they're getting fructose and they have this, they'll start to get these symptoms relatively quickly, and there won't be much they can do about it. Now, of course, you as a nurse can do something about it, right? You can change IVs, IV fluids, and they can heal from this. But it's something to be aware of. It's kind of interesting that we didn't know about this until something went wrong in the hospital. And now, when it happened, I read a paper that happened maybe like 2 years ago. There was a young man who started to have these symptoms, and so they removed the IV, and they talked about his family history, and they did a quick genetic test, and they found out that he and his mother and his sister all had HFI. And they didn't know. They just didn't like beef. They were just a family who didn't have dessert, So the one positive of having HFI is that they tend to have really good teeth. And that's actually medically documented. Very good teeth. Okay. So that's HFI. So you can see these pathways, again, they look a little scary at first, but we can break them down and understand how they're connected. And you can understand how losing just one enzyme can have a really big impact on someone's health and well-being. Okay, so that's that story. Questions about HFI? Questions about albumin C? Anything like that?
I'm hoping the second time I watch it, it'll just make it clearer, and
Yeah, it takes repetition. Repetition is your friend. Okay, I do have videos.
That's what I was thinking.
That are in the course of study and in the cohort. So feel free to watch those as well. Watch them as many times as you need to. And it will come together. It will start to come together. The best way to know if you really understand it is explain it to someone. Explain to someone what happened to this poor Swiss guy, okay? And once you can explain it, then you know you really understand it. So. Okay, well let's move on. We're actually running a little behind here. My apologies, but I do want you to understand that it's really important. Okay, and it will make the rest of this much easier. So, all right. So now let's talk about sort of central metabolism, what we're going to call central metabolism, meaning it's the part of metabolism where everything kind of feeds into it. And this is going to have to do with food stuff for the most part. So glucose, it goes through glycolysis, which is a set of, like 10 reactions. You don't need to memorize those. Be aware of them. And yes, you're welcome that you don't have to memorize them. And you end up with pyruvate. You actually end up with 2 molecules of pyruvate. Pyruvate is going to get turned into acetyl-CoA. And acetyl-CoA might sound a little bit familiar if you've done the lipids task already. Because remember that fatty acid oxidation, so when we break down fatty acid oxidation, also results in acetyl-CoA. So it feeds in here, okay? Then acetyl-CoA feeds into what we call the citric acid cycle or the Krebs cycle. And we'll get into this in more detail in just a minute, but I want to give you an overview, okay? And the citric acid cycle is a big deal. We call it central to aerobic metabolism because for 2 reasons. One, it's really important. So things can be central because they're really important. And so basically without the citric acid cycle, the cell would die. That's how important it is, okay? The other reason it's central is because very important things come before and after it, okay? Let me just give you an indication of that. And I'm going to need more space here. Woo, maybe not quite that much space. Okay. So the citric acid cycle, it's actually going to produce as its product. It's got 8 reactions that are all kind of going around in a circle. And the main thing it's going to be producing here is 3, each time it goes around, 3 NADHs, 1 FADH2. And it also produces a GTP, okay, which can be turned into ATP or used on its own. It's just another kind of energy molecule. But we can't live on just one GTP, right? We need a lot of ATP for this stuff. So the NADH and the FADH2, these are electron carriers, okay? They take up these high energy electrons from the citric acid cycle, and they're going to carry them to the electron transport chain. Okay? And eventually, after the electron transport chain and oxidative phosphorylation, we'll get to that, we're going to make a lot of ATP. Now, the exact numbers here get a little squishy. Some estimates are as high as 38, some are closer to 30. It really depends on a lot of different factors in the cell, but just get an idea, it's a lot. Okay, we can produce a lot of ATP. Keep in mind the citric acid cycle is not producing the ATP, but it's absolutely essential in order for that ATP to be made. Okay, so you have some questions in your task that ask you to talk about a defect, for example, in the citric acid cycle. In that question, you want to connect the dots for the evaluator. You want to show them that you understand why the citric acid cycle is so important for ATP production, even though it doesn't make any ATP. Okay, so you'll start at whatever defect enzyme you choose. This is a hypothetical, by the way. Don't look for a specific disease or anything. Like, this is a hypothetical. You're going to knock out an enzyme in the citric acid cycle, and then you're going to say, what's the effect? What's the effect for the product of that enzyme? What's the effect for the citric acid cycle as a whole? What's the effect for the product of the citric acid cycle? What's the effect for the electron transport chain? Or what's the effect on overall ATP? Okay, again, specific numbers, not important here. Making those connections is what's important. Okay, so there are connections, right? We've got glycolysis and fatty acid oxidation. Also, we eat carbohydrates, fat, and protein usually, right? And so amino acids, when they get broken down, they can actually enter, it depends on the amino acids. Some of them kind of will enter through pyruvate, some of them will enter through acetyl-CoA. You can see how everything's kind of converging, right, on this citric acid cycle. And eventually it's going to enter through this acetyl-CoA molecule into the citric acid cycle. To make those products, you go to the electron transport chain to make ATP. So now we're going to get a little more specific, okay? All right. So if I look at the citric acid cycle, and again, don't get scared. This is just a pathway. It happens to go in a circle, but it's just a pathway. Look at the arrows. What's before the arrows, what's after the arrows. It's all the same. Okay? Yeah, so here's our pyruvate. Don't worry about writing down all the names. There's a million of these diagrams on the web, okay? You'll be able to get the names. I really just want you to kind of appreciate the overall scope of the citric acid cycle right now. So we know glycolysis, that's going to make our pyruvate, right? Pyruvate gets turned into acetyl-CoA, and pyruvate dehydrogenase it's an ACE, it's an enzyme. That's what this arrow is representing, is this enzyme, and it's what turns pyruvate into acetyl-CoA. Okay, it's not part of the citric acid cycle, okay? Some students get confused about that because it's often shown on citric acid cycle diagrams, but it's not part of the cycle itself. So remember, cycle means circle. So whatever's on this circle part, that's part of the citric acid cycle. Before that, it's not. Okay? So when you're choosing your defective enzyme, make sure it's one that's in the circle. All right. Then we've got our acetyl-CoA. Now, acetyl-CoA, these two carbons here are the acetyl part. They're kind of the action end here. Okay. So we've got two carbons available. Oxaloacetate, and yes, you do need a PhD to pronounce all these things. I'm just kidding. Has 4 carbons. 1, 2, 3, 4 carbons. And so when citrate synthase, so here's our enzyme, okay, that's what this arrow is representing. Notice that citrate synthase has 2 substrates. It has oxaloacetate and it has acetyl-CoA. It's going to take both those substrates and it's going to stick them together. So now I've got citrate, which has 6 carbons. Okay, 4 plus 2, 6, right? That's what citrate synthase is doing. And then we just kind of move around the cycle. We've got 8 enzymes here. A conatase is going to take citrate, its substrate, and turn it into isocitrate, its product. Isocitrate now is the substrate for isocitrate dehydrogenase. Really easy names, right? They're so great. They just flow off the tongue. Anyway, and the cycle goes on. We don't have to go through all of the enzymes. You'll get to look at them enough when you're writing your diagram. But the thing I want to alert you to is that it's pretty easy to know when we're making an NADH, and or FADH molecule. And the way to know is to look at the name of the enzyme. A dehydrogenase, okay? It means it's, so dehydrogenase. So it's taking hydrogen from the substrate and it's giving it to the NAD or to the FAD. And along with that hydrogen goes those electrons, okay? So high energy electrons we talked about. So wherever I have a dehydrogenase, I'm going to be making an electron carrier. So I've got NADH here for that dehydrogenase. Here's another dehydrogenase down here. And guess what? Another NADH. And over here, we've got a synthetase. It doesn't make NAD or FADH, but it does make our G-synthetase. Here's another dehydrogenase, and it's making our FADH2. And when we come up here, lo and behold, one more dehydrogenase, malate dehydrogenase, and it's making an NADH. Okay? So the other enzymes are also equally important. Keep in mind, it's a cycle. Okay? Think about a wheel. If I have a wheel, like a bike wheel or something, and I put a brake on it, if I put a brake at the top, will the bottom of my wheel keep spinning? What do you think? Does the rest of the wheel keep going and just that part with the brake stop? What do you think? Oh, everybody's being so quiet. Please speak up. Write it in the chat, whatever you want.
I think it would not spin.
Yeah, good. Thank you, Rosalind. Exactly. We know that from just our physical experience, right? It doesn't matter where I put that brake on my wheel, top, bottom, side. It's going to stop the whole wheel, right? It doesn't just stop that one part. Why? Because it's a cycle. It's a cycle.
Yes.
And it's the same thing with the citric acid cycle. Sometimes students think that, oh, if I knock out a kinase, well, then I can't make isocitrate dehydrogenase, or excuse me, isocitrate, but somehow the rest of the cycle will keep going. We'll just sort of skip that part. And that's not the case. If a kinase doesn't work, then isocitrate dehydrogenase doesn't have a substrate, which means the next enzyme doesn't have a substrate, and neither does the next one, or the next one, or the next one, all the way around, right? The whole cycle comes to a screeching halt. Okay, so keep that in mind when you're doing your task. Okay. All right, so there's our citric acid cycle. Isn't it beautiful? And again, we know that it's making these products, NADH, FADH2, and those are going to go to the electron transport chain. And I have a video on the electron transport chain, so I'm going to do kind of a quick overview here just because we are running out of short on time. But keep in mind, this is all happening in the mitochondria. So here's our mitochondria. It has two membranes. It has that outer membrane that sort of defines its boundary, and then it has this inner membrane that's kind of convoluted. And the reason it looks convoluted like that is because that increases its surface area. And it's in this inner membrane that we have those electron transport chain proteins. Okay, so the more membrane surface area I have, the more electron transport chains I can have, which means the more ATP I can make. Okay, so mitochondria with more folds on the inner membrane, they make more ATP, they win. Okay? They win. Now the citric acid cycle is happening here in the center, in what we call the matrix. It's like the movie, right? Of the mitochondria. And so down here in my little diagram, this membrane that you see, this is the inner membrane of the mitochondria. Okay? On this side, the bottom part, this is the matrix. This is where my citric acid cycle is happening. Okay? And then this is the intermembrane space on the top side. So it's this part right here. Okay? Intermembrane. All right? So it's protected from the rest of the cytoplasm, but it's separate from the matrix. Why do we care about that? Well, remember, think back to your lipids. And think about the way that the membranes form, right? You've got those phospholipids. They provide a very effective barrier. It's very hard for anything to get through that membrane unless a transport protein specifically gets it through. Okay, but membranes are great barriers. And we can use that to our advantage. So here, we've got our Krebs cycle, our citric acid cycle. It's made all this NADH, right, and FADH2. They are floating around the matrix, and when they bump up against that inner membrane, then they run into these proteins, and they're able to deliver their electrons. So NADH delivers its electrons to complex 1. FADH2 delivers its electrons to complex 2. And coenzyme Q10, that's what this thing is here. By the way, the name is a little confusing. It says coenzyme, and so you think, oh, enzyme, it must be a protein. It's actually not. If something's a coenzyme, it basically means it's a vitamin. It's something that proteins can use to do their job, but it's not a protein itself. Okay? It's just a big, flat molecule is really all it is. Okay? So don't be fooled by the name. Okay? All right. Now, coenzyme Q10 will pick those electrons up from complexes 1 and 2, deliver them to complex 3. Cytochrome C, another coenzyme type thing, takes those electrons up, delivers them to complex 4. And now, we've got to get rid of this electron. Why? Well, remember they started out as high energy. When NAD and FADH2 gave them to the electron transport chain, they were high energy. So you can think of them as being up here at the top of the staircase. And every time they get transported around, right, remember this is the electron transport chain, so we're transporting electrons. Every time it does that, it drops down an energy level. And we can use that energy to do work. And we do. These complexes actually harness that energy, and they use it to pump out these H pluses. These are hydrogen ions, or we also sometimes just call them protons. Okay? They're pumping out these protons. They're using the energy from the electrons to pump out these protons. Now, by the time the electron gets to the end of complex 4, it's like out of energy. Okay? It doesn't have a lot of energy left. So we just need to get it out of the way so it doesn't back up the system. Right? If it just stayed here, well then I would get backed up, right? Mm-hmm. And so oxygen comes in, and oxygen says, I'll take that electron. Come here, electron. And together they'll actually form water with some other hydrogen, of course. And this right here, I can't even emphasize enough. It's so important. This is the whole reason we breathe. This is the whole reason we need to breathe. Right here is so that we can move those electrons, so that other electrons can move through, and we can pump out these protons. Okay? The whole reason we breathe. Oxygen is what we call the terminal, meaning the last, electron acceptor. It takes those electrons, makes water, and the system can keep going. Okay? Okay, great. So this in green here, this is what we know as the electron transport chain. That's what's transporting the electron. What about this yellow guy over here? I mean, so right now we've transported electrons, we've pumped out some protons, so now on this intermembrane space side we have just an enormous amount of protons. Okay, great. Now what? Okay, now what? Well, I want you to think about, in my video I used an example of a hydroelectric dam, but I want to use a different one for variety. Think about Black Friday. Okay, think about Walmart on the morning of Black Friday. There's Walmart. I'm looking down on Walmart. Okay. Here's the door. And I've got like 1 million people out here, right? Just tons and tons of people just waiting for those doors to open, right? There is inherent energy in having a huge crowd of people on one side of the door and virtually nobody on the other side of the door, right? Naturally, those people want to go in, right? They want to go in through that door. And when they do it with energy, right? They do it with gusto. They're like, yeah, let me in. I'm getting that flat screen TV or whatever. And so they're flowing in through the door in a controlled way, right? They can't go in through the walls. Whoa, whoops. They can't go into the walls. They have to go into the door. They are controlled. But when they go through, they have a lot of energy. Now imagine I put like a, I don't know, like a turnstile. You know those little turnstiles where when you go through it, like an amusement park, it turns, or like in a metro or something. It turns as you go through. If I had a genius machine, I could harness that energy to do some other work, right? I could make a little machine that runs off of people walking through the door of Walmart. Well, our cells have already done that. This yellow protein here, I don't know why they have a 5 here, it's called ATP Synthase, meaning it synthesizes or makes ATP. This is the door. This is the door into Walmart, okay? All of these protons really want to get back over to the matrix because they are crowded on the intermembrane space side. So this ATP synthase lets them through a few at a time, controlled way, and when they come through, they are flying through, right? They are going so fast. And the ATP synthase can use that energy to make ATP. And as we mentioned before, when we say make ATP, what I mean is they take ADP, the diphosphate, and it adds a phosphate to it to make ATP.
Yeah, to make ATP.
That's what it's doing. Now, ATP synthase, sometimes it gets lumped into the electron transport chain, just for convenience sake. But the truth is it's really not part of the electron transport chain. Instead, it's what we call oxidative phosphorylation. Why? I don't know. Because we want to make it hard. I don't know why. But phosphorylation means I'm putting a phosphate on something. Okay? So we're putting a phosphate on ADP here. We're making ATP. And so that's what we've got. That's what we're doing. So when you're talking about the electron transport chain, your task, just be aware. It's the electron transport chain, yes, but it's also ATP synthase. It's this oxidative phosphorylation. And together, they make ATP. Okay? Yeah. Yep. So this is right here, oxygen, right? This is why we breathe. Our body can do, so this is aerobic metabolism, right? Aerobic meaning it involves oxygen. This is aerobic metabolism. Okay. Now, I should mention we can do, for a short period of time, anaerobic metabolism. We can hold our breath, right? We can hold our breath for even a couple of minutes and not die. Our cells can do anaerobic, no oxygen metabolism for a short period of time. But not for too long, okay? Not for too long, because it just doesn't make enough ATP. Let me show you an example of that. This is what we call the Krebs cycle. And there's really two kinds of cells that use the Krebs cycle. One is a red blood cell. And the reason a red blood cell uses it is because red blood cells are really just bags of hemoglobin, right? They don't have a mitochondria. They don't have any mitochondria. They're just bags of hemoglobin. Now, they still need a little bit of energy to survive, to maintain their membranes and things like that. And so what they can do is glycolysis. And also, we want red blood cells to run anaerobically because they carry oxygen. We want them using their cargo, right? Bad idea. Yeah. We don't want them to do that. So they do everything anaerobic. They can do glycolysis because it doesn't require oxygen. But that's all they can do. So they can get 2 ATP out of glycolysis. And it does make pyruvate, but see, pyruvate, if you're in an anaerobic environment, will get turned into lactate. Okay, so it makes lactate. The cell can then release lactate into the blood. It's going to go to the liver. And remember, the liver is like this pro at gluconeogenesis. It's storing lots of stuff. It has a lot of energy. And so it's able to input. It costs the liver 6 ATP to take those 2 lactate molecules and turn them back into glucose. But that's okay. The liver is like Uncle Moneybags. It's got it. It's covered. It's okay. And so then it can release that glucose out into the blood again, and now our red blood cell can take it up, get 2 more ATP, release the lactate, and the cycle continues. Another type of cell that will use this would be a muscle cell if it's gone anaerobic. So if I'm chasing the bus, right, and I'm running really hard, I'm totally out of breath, but my muscles have to keep going, they'll go anaerobic, which means they'll do glycolysis to get a little bit of ATP so they can keep working for a little while, but they can't do it forever, right? And we all know that feeling of lactic acid burn, right, in our muscles when we've done that, right? It produces that lactate. Same thing. Then it will hopefully release the lactate, and that will go to the liver. The liver will turn it back into glucose, and eventually the muscle can take up some of that glucose and store it, okay? The muscles also use the Krebs cycle. They use it on a much more short-term basis than the red blood cells. But muscle cells take a lot more energy than red blood cells do, so often when we talk about the Krebs cycle, it is talked about in terms of muscle cells, because in terms of the volume, they use it more. Okay, so that's how it works. We make 2 ATP for the cell that needs it, and then the liver takes care of the gluconeogenesis, takes the glucose again, and they go around the cycle. And the reason that this works is because it's in two separate situations, right? It's in two separate areas. And in your task, you're actually asked to talk about a hypothetical. And I just want to, like, set up the hypothetical for you, okay? I'm not going to do all the work for you. You got to do some thinking, but let's just set this up. The hypothetical, just so you understand, is they're saying that the entire Krebs cycle, that means glucose going to lactate and then back to glucose, they're all in the same cell. So the same cell is doing glycolysis and getting 2 ATP. It's also doing gluconeogenesis. Gluconeo, we'll just leave it at that. And it's costing the cell 6 ATP. And so the question in your task is this going to work? Think about a hypothetical where the entire Krebs cycle is in a single cell. What would be the consequences, right? If I'm gaining 2 ATP, but I am spending 6 every time I do this cycle, what will happen to the pool of my ATP in that cell? And, you know, how important is ATP to cells, right? What would happen to the cell eventually? Those are things I want to think about when you're answering this hypothetical. Okay. All right. Let's see. I'm trying to think if there's anything else I can connect with this for you. Oh, sometimes in exercise and things you'll hear the term oxygen debt. Has anybody heard that before?
Oxygen what?
Oxygen debt.
I don't know if I've heard of it.
No, I mean, I used to be a sprinter, and so they would talk about this a lot. Because you're like, you know, pushing your muscles really hard. And you build up what they call an oxygen debt. That's basically the Krebs cycle. It's because, you know, you're creating all this lactate because you're working anaerobically. You don't have oxygen.
Oh, okay.
And to turn that lactate back into glucose, back into something usable for your cells, it costs a lot of ATP, and so they call it debt, right? It's like it's an oxygen debt because I didn't have enough oxygen. I'm creating this like energy debt. But that's part of what that's referring to. So you're using, it costs ATP. Like if you look at the Krebs cycle, when the cell turns glucose into lactate, it gets 2 ATP, but it costs 6 ATP to get it back to glucose. So the Krebs cycle runs at a negative 4 ATP, right?
Oh, okay.
And so the more I have to use it, meaning like the more anaerobic I've made my muscles, the more ATP I'm losing, right?
Okay.
Which, if you're trying to lose energy, is not such a bad thing, right? But if you're not trying to lose energy, then it can be. So,
okay.
It's nice. I'm trying to think if there's anything. There's one other thing. When you are asked to talk about coenzyme Q10 in your task, this is just like a task.
When you're asked to talk about what?
Coenzyme Q10. So there is a question on coenzyme Q10 here in the electron transfer chain. Read the whole question because it's not just saying what's the role of coenzyme Q10. It's asking what is its role in ATP synthesis, which means you're going to have to explain the whole process. Okay, to get from, you know, coenzyme Q10 here and its role all the way to here, ATP synthesis, you're going to have to explain the whole electron transport chain and oxidative phosphorylation. Okay, so just be aware of that. It's really just in the context of a larger explanation. Okay. And that's all I got. I hope that was helpful. I hope that was helpful.